Hello everyone, welcome to my presentation, and this is Nuhu from icddr Dhaka Bangladesh, and today I'm going to present about the performance of different on-site sanitation technologies, removing uh, pathogens from the urban low-income neighborhoods of Dhaka. So without further ado, let's get started. So as many of you know that Bangladesh has made tremendous progress on the latrine coverage and reducing open defecation. And in 2016, government of Bangladesh declared the country as a open defecation free nation. Despite the success, the recent sheet flow diagram showed that more than 97% of the sludge directly going to the fecal waste directly going to the environment without treatment, and we expose a huge human health exposure. WHO also recently exposed substantial knowledge gap on the environmental pathogen exposure and health impact. There are also a limited understanding of pathogen removal in on-site sanitation system, particularly removal of different types of organisms type. For example, virus, bacteria, protozoa, and helminth. So as a result, sanitation investment decision-making are really informed by consideration of which option will best to reduce the human health risk. So to explore some of these questions, we identified the frequency and concentration of target pathogen in range of environmental sample type across different seasons and different volumes of samples. We also explored the influence of latrine coverage and the septic tank coverage on pathogen concentration in drain and also the surface water. We also explored the relationship between the level of general fecal contamination in each sample type and the likelihood of the pathogen detection in the collected samples. So we conducted a cross-sectional study from April to October 2009 in 2 seasons. We conducted the study in 2 low-income communities from Mirpur. From study site A, we collected samples from septic tank, different layers of septic tank, different points of open drain, and also the surface water. From study site B, we collected samples from anaerobic baffled reactor. So this is how the study site A looks like. Study site A had 4 roads, ends up in a canal, and you can see the septic tanks and on-site sanitations is located in these blocks. We collected drain sample from end of the drain and the canal sample from the center of the canal of each drain. We also collected samples from sediment samples from the center of the drain. We also collected samples of flood water randomly from different households during the rainy seasons. So we use septic checkers. So this is the device that can measure the different layer of samples like sludge, layer of sludge, say layer of supernatant, and also scum. And from septic tank ABR, we have collected 3 different types of sample from each system. For example, the sludge from the bottom of first chamber, supernatant from the liquid portion of the first chamber, and effluent from the outlet of the chamber. So we also collected samples from grab, drain sample, surface water, canal, and also the flood sample. So we also collected 2 different types of different volume of sample to compare the detection efficiency and also the concentration comparison. From the drain sample, we have collected drain sediment from the center of each drain. So let's discuss about the indicator organism and other pathogens we have selected. So E. coli was the indicator organism we used for this study. And for the virus, we have selected norovirus, 3 bacteria, Vibrio cholera, Shigella, and Salmonella typhi, and also 2 protozoa, Giardia and Cryptosporidium. We conducted in-depth laboratory method to quantify E. coli. We use IDEX quantitative technique. And for the pathogen detection, first of all, we concentrated the pathogen using the PEG precipitation technique. And after DNA extraction, we did TACMAN singleplex real-time RT-PCR. The equivalent genomic copies were estimated by interpolation of the mean CT value in the corresponding standard curve and the dilution factor for each sample type. Absolute concentration was explored in log equivalent genomic copies per 100 ml for each water samples. And the sediment sample was explored by per gram. So this is the results, like frequency tables of each type of samples for each type of pathogens. So you can see throughout the environmental samples, except for drain sediment and the septic supernatant, most of the samples are highly contaminated with different type of multiple type of pathogens. So if you see the effluent that directly goes to the environment from ABR and also the septic tank, so this is these pathogens are directly going to the environment and mix with the drain and surface area. So we also see the concentration was also high for Vibrio cholera, Shigella, norovirus, Giardia, and also high concentration of fluoride was also found from this set. This box plot is also showing the difference between the dry versus wet season sample concentration. The wet season sample concentration was significantly higher than dry season concentration for Vibrio cholera and Shigella. E. coli was also highly concentrated during the wet season. So when we compare the two different types of volumes, so pathogen detection were more frequent in large volume of sample compared to 400 gram. Percentage of sample detection with positive pathogens was also more in ultrafiltration samples compared to graph samples. But the pathogen concentration reduced by ultrafiltration method, but increase in the graph samples. So this graph is showing the latrine coverage of the septic tank coverage of different drains. So I would like to highlight this 90% coverage versus no coverage, drainage coverage here, septic tank coverage in this here. So there is a reduction of certain pathogens after the 90% coverage of septic tank, but like overall concentration didn't reduce much after this investment or after this installation of this septic tank. We also found like trend of concentration from between, see the association between the E. coli and other pathogens concentration. So when the concentration of E. coli increase, there is a possibility of concentration of norovirus, Vibrio cholera, Giardia, Shigella, and Salmonella typhi also increase. So there is this increased trend of pathogens with the increase of E. coli concentration. So in conclusion, high frequency and frequency of pathogen detected in almost all the locations of the sample types with high concentration of pathogen in effluent, drains, surface water, and flood water. Septic tank didn't make much difference. Drains in road with high septic tank coverage had similar type of pathogens and E. coli concentration to road with lower coverage of septic tanks. Wet season has more concentrated pathogen compared to dry season. So what is the implication and for the key stakeholders from this study? That we found that the key and the way we currently design these on-site sanitation options is not effective to remove pathogens. So as a result, this could be a major threat for the disease. Seasonality is an important issue, so we have to consider these during construction and during the assessing the efficacy of this on-site sanitation technique. Incremental incremental improvement of these systems are possible. For example, we didn't actually explore the desludging technique a lot because the information is not so available. So we have to explore this in the future. And also future study is important to explore the more geographical and rigorous research to explore more data for this type of analysis. So we have published this in 2000 recently, and last month this paper was published in International Journal of Hygiene and Environmental Health. Another modeling main paper, the primary outcome paper from this study, is also accepted and revision submitted to the same journal. So I'd like to thank all of my collaborators and all of my colleagues here. Thank you very much, and I'm happy to take any question. Thank you very much.